Hello, my name is James Claiborne, and I'm here to talk to you today about a research proposal that I have thought of. It is called A Study of the Endocannabinoid System, Comparison of the Effects of Endo and Exocannabinoids. Endocannabinoids being those that are produced endogenously, exo being those that are produced exogenously. And in this study I asked the question, would endocannabinoids elicit similar states as exocannabinoids if the concentration was increased? This topic is of interest to me because the exocannabinoids are increasing in prevalence in communities such as the one I grew up in, and I don't know much about them. So why do I want to study them, you ask? Why should I care? Well, for starters, exocannabinoids can be found in plants, with one of the most popular being cannabis. Cannabis has raised controversy and social concern about its recreational and medicinal use and applications. Understanding more about its effects can address these concerns, at least hopefully. And since it has applications in neuronal and systemic pathologies, understanding it is paramount to the health sciences. The more we know, the more we can do. Regarding the endocannabinoid system, there's not very much circulating around it, knowledge-wise. Part of the reason why I do not know much about the topic is simply because there's not much known. The system has been studied for about 50 years, but the biosynthetic pathways are still relatively uncertain. What is known about the system, although, is it is mainly inhibitory, and it is involved in energy balance and metabolism. Also, the endocannabinoid system has been suggested to have central roles in appetite and hunger. These central roles that the endocannabinoid system are suggested to have to play a part in are facilitated by the cannabinoid receptors CB1 and CB2. These are intron-less G-protein coupled receptors that can act on GABAergic and glutamatergic neurons. CB2 is mostly found on immune cells and modulates immune function. CB1 is found mostly in the brain and spinal cord. Given these regions they are mostly found in, CB2 is dubbed the peripheral cannabinoid receptor, CB1 the central cannabinoid receptor. Being dubbed the central cannabinoid receptor, it is among the most abundant G-protein coupled receptors in the brain. It is expressed in high levels in the hippocampus, cerebellum, and limbic cortices. Also, CB1 is highly expressed in CNS neurons that regulate feeding, energy expenditure, and reward. Activation of the CB1 can cause inhibition of calcium, lower cyclic AMP levels, and activate calcium channels. In addition, they are constitutively active, meaning that they are always on. So every receptor needs a ligand, and some of the currently known ligands of CB1 are anandamide, 2-arachidonilamide, delta tetrahydrocannabinol, cannabinol, and cannabidiol. Unlike neurotransmitters, these are synthesized on demand and not stored in transport vesicles. Also, they act in a retrograde fashion. They retrogradely suppress the presynaptic neuron and inhibit the function of calcium, which results in the inhibition of neurotransmitter release, staying true to its inhibitory nature. To address my question, there are two parts. The first is a microscale experiment. It requires that cells expressing CB1 are cultured. Then these cells will be introduced to varying concentrations of anandamide. Assays will be performed to screen for physiological changes in the treated cells and compare the untreated and exocannabinoid treated cells. Tests that can quantify cells of intra and extracellular concentrations of calcium and potassium can provide data about what is actually occurring physiologically in these cells. Then, using the results of the microscale experiment, giving that certain concentrations of endocannabinoids elicit significant effects, we can inject mice with the effective doses of anandamide and compare it to untreated and exocannabinoid-treated mice. Since mice will be used, another dimension of observation can be addressed. We can look at their behavior. The behavior of the mice can be observed and compared to determine if the states induced by exocannabinoids can be achieved with excess endocannabinoids. We can also screen for the physiological differences just to make sure they are the same. So what can we find out from this study? We can learn about the role and importance of proper endocannabinoid regulation. It also can give insights as to what is occurring when exocannabinoids are ingested. Some interesting questions that can arise will come if no similarities are observed. Since the accepted active ingredients in cannabis are THC, CBD, and CBN, which are all agonists of the same CB1 receptor, no similarities would suggest that those are not the most important compounds acting on the subject when they are using exocannabinoids. In summation, the endocannabinoid system is a relatively unknown system that demands our attention. This study can help further the understanding of this pathway, and understanding this pathway can lead to many different beneficial outcomes. The ECS may introduce treatment options associated with neuronal pathologies. For example, if its main function is inhibitory, maybe disorders that have excitatory complications can be addressed. This is important information that may be key to saving lives. It is just waiting to be discovered.